I don't ever mind you guys reminding me of that. The reason I'm not going to lecture on that is because it's essentially a list of terms. It gives you the descriptive text and lets you know where it's at. I think you would get far, far more reviewing that yourself than you will with me standing up here, putting up a slide, pointing my finger and saying the same thing. Is everyone okay with that? Now, I also want you to make sure that you feel free to ask me for any help you may need. This is not me just throwing something off on you and not being willing to teach it. I just think you'll do better by yourself learning it than you will with me lecturing. Yes, ma'am.
Could you repeat which chapter it is on?
1.6.
Chapter 1, Section 6, 1.6, and then Chapter 1, Section 8. I believe that's on pages 28, 29, 31, somewhere in that range. I'm not a big fan of memorizing lists. That's very, very basic information. But unfortunately, as you're going to see, anatomy is a lot of very basic information. Don't ask if you need to know that for the test. The simple fact is you need to know that for your career. You need to be able to walk up to a skeleton and know that the axial skeleton is the head, the neck, the torso, the trunk. The appendicular skeleton will be the limbs. You see where I'm going with this? Superior means above, inferior means below. Proximal versus distal. These are just simply terms that help you find your way around the body. Is everybody clear on that? Had a couple of people ask me, and I just wanted to make sure that you know that that is not only material that you need to know, it's expected you do know it, okay? If I use those terms and you don't know what they mean, don't hesitate to ask, okay? I don't expect everybody to just say, oh, I read it once and I know everything on there. But I also don't want to be telling people the difference between superior and inferior the last week of class. That's why we're here, to learn it all. Is everybody okay with that? All right, so next up, we are going to finish our little sojourn into chemistry. And is there anybody who has had difficulty with chemical equations, or do y'all feel comfortable with that now? I see most of you nodding. It's something, again, it's a little bit more of a digression than I like to make, but it is relevant to what we do and what we study, because when we start talking about ion levels, when we start talking about pH and those types of things, you need to know what it means, okay? Is everybody with me on that? So, main thing I want you to remember, next up, we're going to talk about acids and bases, electrolytes, and salts. Acids, bases, electrolytes, and salts. And for the body, the main one of these you're going to be concerned with is going to involve pH. Yes, ma'am.
Could you repeat the second one?
Acids, bases, electrolytes, and salts. Section 2.4 in your textbook. Acids and bases, what are they? Well, to make a long story short, acids are proton donors. Bases are proton acceptors. May seem like a simple definition, but it's probably the best we've got out there. And I'll show you what happens in terms of the chemistry. hydrogen atom is simply a proton with an electron in orbit, right? We all agree on that. When hydrogen loses an atom, or loses an electron, all that's we're left with is a naked proton. Does everybody see that? That's all an acid is. It's a proton donor, because essentially it's just a proton. A base is a proton acceptor. It picks those up. And that's all there is to it. Now, we measure acidity or basicity as pH. pH measures only weak acids and bases. It measures only weak acids and bases. Why is that important? Well, it's important because people seem to have this idea that if you're exposed to acid, it's something like out of a Bugs Bunny cartoon, where the flesh literally just melts off your bones and you're left with nothing but the bones, right? Only thing that can do that are very, very strong acids. And even then, you generally have to have a lot of heat and agitation involved in it. Is everybody clear on that? Vinegar is not going to burn your skin. Vinegar is a weak acid. Is everybody clear on that? I want to be very clear. Basically, if you can measure your pH, you are not dealing with a strong acid or a strong base for that matter. So what exactly is pH? It is a measure of the concentration of protons in a solution. It is a measure of the concentration of protons in a solution. Everyone kiss back.
What was the last word? I'm sorry.
Solution. Everybody okay? Now, I want to introduce you to the concept of a logarithmic scale. A logarithmic scale. Essentially, this is an exponential scale. An exponential scale. So every time, if you're looking at a logarithmic graph and it's base 10, every time you move up, you're not doubling, you're multiplying by a factor of 10. Does everybody understand that? So basically, you're using scientific notation. One times 10 to the first is 10, right? Doesn't equal 20, it equals 10 times 10, which is, everybody see how this goes? You can also do the same thing with a negative scale, okay? And what do I mean by that? Decimals. Right? Everybody see how that goes? Do you need me to make more?
I just need you to explain it again if you don't mind.
If it's a negative exponent, it means you're dividing by 10. If it's a positive exponent, it means you're multiplying it by 10. Everybody clear? Reason I'm telling you this is because I got all the way through my doctorate knowing that a pH above 7 was a base, a pH below 7 was an acid, but I couldn't tell you what the number meant. I couldn't tell you what it's actually measuring. So that's what we're doing here. Is everybody okay with that? If you ever measure the molar concentration of something, you put it into brackets. Concentration can be a lot of different things, right? It could be mass per volume. It can also be molar. Do y'all remember what a mole is from chemistry? Oh dear, how to explain molarity.
I didn't even take chemistry.
How many of you have never had chemistry? Oh boy. Okay, a mole represents the ratio of atoms in a component. That's a good working definition. It represents the ratio of atoms in a compound. So for example, H2O, right? There are 2 moles of hydrogen to 1 mole of oxygen. Does everybody see that? The molar mass of an element, you may want to get this one down. The molar mass of an element corresponds to its atomic weight. The molar mass of an element corresponds to its atomic mass. Yes, ma'am.
I'm sorry, can you repeat that one more time?
Sure. The molar mass of an element corresponds to its atomic mass.
Thank you.
You're welcome. So if you were looking at your periodic table, you're going to see that hydrogen has an atomic mass of 1. Everybody see that? What's the atomic mass of oxygen? Somebody look it up for me.
Would it be 16?
Huh? 16? Yeah. So 16 grams of oxygen is 1 mole. 1 gram of hydrogen is 1 mole. Does everybody see this? You understand now? If we have something like salt, what's the atomic mass of sodium? Somebody tell me. Na. First column, Na. 23, okay. And what is the atomic mass of chlorine? 35. So 23 plus 35 is 58, right? Does everybody agree? So to have what we call a 1 molar solution of salt, we would take 58 grams of salt, dissolve it in 1 liter of water. Thumbs up if you got it. Okay. Does everybody see where I got 58 grams from? Sodium chloride, NaCl, right? Sodium is 35, or it's 23, and chlorine is 35. You with me, Desiree? You agree with that, Piper? Okay. Those two together come to 58. The molar mass of salt is 58. And since this is the nice metric system, we're going to use grams, right? Now, if I dissolved 58 grams of salt into 1 liter of water, that's going to be fairly concentrated, isn't it? And we call that a 1 molar solution, a 1 molar. Molar is a measure of concentration. Let's assume that instead of dissolving it in 1 liter, I dissolved it in 10 liters. That's going to be a lot less salty to the taste, isn't it? It's not as concentrated. Does everyone see that? In that case, it would be a 0.1 molar solution, a 0.1 molar, because it's not as concentrated. You with me, Annika? Is a review for you? Is this just a review for you?
Yeah, I guess, yeah.
Don't go to sleep on me, Kai. This is one of the reasons I don't like talking chemistry. It's hardcore. But does everybody see how it's a measure of concentration now? Well, if you are measuring the concentration, the molar concentration of protons, you can use the same numbers. Does everybody see that? So let's say I'm trying to measure a concentration that's that small. That's a painful way to write that number, isn't it? It's easier if I write it like that. Does everybody agree? But without getting into the nitty-gritties of terminology and why they do it this way, it's easier to put a P in front of it. That lets you know it's a logarithmic scale and a 7. So does everybody see what that means? When you see a pH, it means there are this many hydrogens in 1 liter. And now I think you see why people prefer pH, right? If we had a pH of 14, which is the other end of the scale, I would have 13 zeros followed by a 1. There's not a lot of protons in that, are there? That's because it's a base. So everybody got that? pH only measures weak acids or bases. Strong acid can have a pH equivalent in the thousands. If you didn't understand all this, it's okay. I wanted you to know, though, where the numbers come from, what they're actually measuring, and what it actually means. Everybody see that? pH is very important in medicine and normal function. Most enzymes have a very narrow range of pH at which they will function. Most enzymes only have a very narrow range of pH function. The pH of blood is slightly basic. Typically about 7.4. If the pH goes down, you can get a phenomenon called acidosis. Acidosis. It's just an acid buildup in the blood. To prevent that, the body uses substances called buffers. Buffers. A buffer is simply a substance that supplies needed protons or accepts excess protons. It will either donate a needed proton or it will accept excess protons. Did that help you all? Y'all do know the normal range of pH, yes? You've been exposed to that at least? It's okay if you haven't. pH of 7 is neutral. pH of 7 is neutral. That is the pH of water. That means even in a glass of water that's just sitting still on your desktop, a very small amount of it will have broken down into protons. Everybody see that? Anything above 7 all the way up to 14 is a base. A base. They're sometimes called alkaline elements. A base. If you've ever used Drano, you've used a base. That's pretty much sodium hydroxide poured right down your drain. So everybody got that? Anything below 7 down to 1, or technically you could even go as long as it's above 0, is an acid. It's an acid. So if your blood pH is 6.8, it's slightly acidic. Everybody see how that goes? You understand pH? I'm not going to go any more into the chemistry of it, but I want you to understand what it is it's measuring and why it's important. Because in an acidic environment, those excess protons want to react with something, don't they? Generally, it's not a good thing when you have random chemicals reacting with your body structures. Anybody here ever gotten an acid burn? A few of us, yeah. It's not pleasant, is it? I think I still got a scar right there where I was doing analytical chemistry and we were dissolving iron ore in nitric acid and somebody hit my left elbow. It spilled over onto me, and since it had iron ore dissolved into it, I've got a little rust spot. You can laugh, it is kind of funny. Good story to tell people. It burned. Released heat. Okay, any questions on acids? Electrolytes. Electrolytes. What are electrolytes? What are salts? Well, electrolytes are simply substances that dissolve in water. Substances that dissolve in water. So is an acid an electrolyte? Yes, it is, because it dissolves in water. Is a base an electrolyte? Yes, it is, because it dissolves in water. Everybody see that? A salt is simply the product of a reaction between acids and bases. It is simply the product of a reaction between an acid and a base. If I took a solution of hydrochloric acid and I mixed it with a solution of sodium hydroxide, I will neutralize it to pH of 7, and if I boil the water off, I'll be left with table salt. Everybody clear on that? Electrolytes are, by definition, ions. By definition, they are ions, whether or not they're positively or negatively charged. If you look on page 40 in your textbook, section 2.4, there's a pretty good write-up of what we just talked about. Everybody got that? Did we go over the definition of a solution, a solvent, and a solute?
Yeah.
Okay. So Why is this important for your cells? Well, basically, the cell is just a balloon or a bag filled with a solution. We call that cytoplasm. Cytoplasm. Yes,
Can you
ma'am.
repeat that? I'm sorry. Can you repeat that?
Sure. The cell is essentially just a bag full of an aqueous solution that we call cytoplasm. What you need?
No, nothing.
I'm sorry?
Nothing. I was just saying I heard golf whenever I was studying.
Aqueous, AQUEOUS, is just another term for in water. It is a solution in water. In other words, water is the universal solvent, so you're going to have a lot of water in your cell. Everybody with me? Other things you're going to have in solution are going to be a variety of ions. Typically, those will be various salts that have dissociated. So you're going to have a lot of potassium, sodium, phosphate, hydroxide. You get the idea. Other things you may have are gases that are dissolved in the liquid. Oxygen. Carbon dioxide. Nitric oxide. Carbon monoxide. A couple of those are organic, you know that? They contain carbon, right? The word organic in chemistry just means carbon. But a lot of those are inorganic. If you take a look on page 41, your book does a pretty good job of laying out all the various inorganic things you found. Any questions? Huh?
So with like measuring pH, we only do it in liters. With measuring like pH and like trying to figure it out, it's typically only in liters.
Molarity is only in liters. pH is just one way to measure it.
Okay.
Going back to dissolving 58 grams into, we could say that is a 58% solution of salt, couldn't we? See where I'm going with this? It's just a convenient measure.
Okay.
One of the reasons I always ask people who've had chemistry and try to give you some crash course in it in a couple of days is because, like it or not, biologists have to have a working knowledge of chemistry. You're probably more used to thinking of percentages like pounds per gallon or something like that. But this holds across all branches. So molarity is a useful way to do it. Good question though. And if you don't like the metric system, I feel sorry for you because you will need to know it. Because essentially science, it runs completely on a metric system. Is everybody with me? Are there any questions over the chemistry part? Everybody knows what organic chemistry is, the chemistry of carbon. Inorganic chemistry, everything else. You know the difference between an ion and an isotope, right? An ion is a charged atom. An isotope has a different atomic mass. You remember chemical bonding, right? Ionic, covalent, hydrogen. Everybody can define polarity, yes. If you don't know these terms, I would strongly suggest you study this weekend because you will need to know them. Everybody cool with that? Any questions? Okay, that's it for chemistry. Now we're going to jump back to the cell. The cell. I talked a little bit last time about the Hayflick limits, different types of stem cells, why regulation of cell growth is important, right? You want and need to grow. You want and need to be able to repair damaged tissue. But you don't want to turn off the control and just keep growing. While immortality sounds great in principle, we all saw that it can actually lead to cancer in practice, right? Everybody understood all that? So what we're going to talk about now is the cell itself. The basic unit of human life is a single cell. Every one of us began as that. By the time it's all said and done, you've got anywhere from 30 to 50 trillion cells in your body. What will you find in each cell? Well, to start with, we're going to talk about organelles. Organelles are those tiny structures in the cell that have specific functions. The one that most people say is the most important is the nucleus. The nucleus. In high school bio, they like to call it the control center of the cell, but in reality, it's just an information depository. It's just like a library. It's where you keep the master plan. It's surrounded by the nuclear membrane. The nuclear membrane controls what goes in and out of the nucleus, not the cell, the nucleus. So it is very, very important in regulating gene expression.
Did you use this on a attendance paper?
Huh?
Did you use this on a paper for attendance today?
I haven't done it yet. I knew some people would be coming in later, so I didn't want Veronica to feel like she was being singled out. Thanks for coming. I'm not being sarcastic. I'd much rather somebody be here late than not show up at all. I have recorded it, so I'll be posting it in case you need it. See, when you let me know in advance, I know how things go. Amazing, sir. Get here. So back to the nuclear membrane. It regulates gene expression. It helps regulate gene expression. Because if you're controlling access to the DNA, you're controlling pretty much what goes in and out. You all do know what gene expression is, yes? DNA, RNA, protein. DNA gets transcribed into RNA, RNA gets translated into protein. That is a gross oversimplification of it, but this isn't a cell bio class. Oh, coming away from that, you will have a structure that is a series of membranes, a network of membranes. This is called the endoplasmic reticulum, the ER. And henceforth, we will always use the term ER. Is that okay with everyone? Every single cell bio class I have ever seen or taken or taught, it ends up being used. ER comes in two flavors, smooth and rough. my freshman in my very first class used to say, smooth and chunky. The rough ER, the membrane is studded with structures called ribosomes. A ribosome is where the RNA gets translated into a protein. It's where the RNA gets translated into a protein. So the rough ER is primarily going to be involved in protein synthesis, protein assembly, the smooth ER is going to be where you're going to have lipid synthesis. Next up, we've got the Golgi, Golgi. Golgi body, Golgi apparatus, the Golgi, I've heard it called all kinds of things. This is basically like an Amazon fulfillment center. Y'all laugh, but it is the best example I have seen. You order something off Amazon, somebody goes in and pulls it off the shelf, right? They then find the appropriate container. They might put some padding on it, and they'll put in a little slip, and then they'll seal the package, won't they? Then they address it, and they throw it on the truck. The Golgi basically does the same thing, except instead of Amazon, it is using molecules. So if you are secreting a hormone, it gets made in the smooth ER, goes to the Golgi packages it, puts it into a vesicle, goes to the plasma membrane, pops it out into the bloodstream. Packing and shipping, if you wanted to sum up the function of the Golgi. Cytoskeleton, C-Y-T-O, cytoskeleton. And anytime you see that prefix, cyto, it means cell. Everybody with me? It's basically like it sounds like. It's just a network of tubes and fibers that hold the cell together and give it shape. It gives the cell support, gives it shape. I mentioned the cytoplasm. It's just the liquid in which everything is floating or dissolved. Mitochondria. Mitochondria are interesting. They actually have their own DNA. And I know in high school they always call it the powerhouse of the cell.
You said they have their own DNA? You said the mitochondria has their own DNA?
Yep. Probably bacterial in origin. But don't worry, we won't go into that. What is the function of mitochondria?
It creates, I believe they're mRNAs for
It creates
the cell
what?
to use.
ATP.
ATP, I got it wrong.
ATP, adenosine triphosphate. ATP, or adenosine triphosphate, is the energy currency of the cell.
Can you say that one more time?
The energy currency of the cell. Back in the dark ages, way, way long ago, before you were born, there was this thing we called cash. And you could use cash to pay for things. You could walk into a convenience store and get yourself a candy bar. You could hand them a $20 bill and go pump some gas. You could even use it to pay your water bill or your taxes, couldn't you? I know everybody does everything electronically now. Uses a cards or touch tap your phone. I haven't figured that one out yet. I'm afraid to try it because I think I'll be linking my bank account to every electronic device in Somerset. But you see what I could use cash for? What is it useful for? Anything I want to pay for, right? If I want to buy something physical, I can buy it with cash. If I want to buy a service, I can pay for it in cash. If I want to pay a debt, I can use cash. ATP is the same way for the cell. It's used for everything that requires energy. You want movement? ATP. You want to bring something in the cell? ATP. You want to make more DNA so you can divide? ATP. You actually want to divide the cell? ATP. Now, I think you see why the mitochondria are of critical importance, right? Next up, the cytoplasm. The cytoplasm.
I thought you were gonna do the cytoplasm.
Oh, okay. Lysosomes and peroxisomes. Basically what these are just a membrane enclosed space. And it's where the cell stores like digestive enzymes. So if the cell takes something in and it needs to break it down, it will stick it into a lysosome. The enzymes in the lysosome will break it down. Everybody clear on that? So if a white blood cell engulfs a bacterium, it'll send the bacterium to a lysosome, it'll break it down. Peroxisomes are where the cell sticks dangerous substances. Peroxide, for example, is very reactive. You don't want it floating around in your cytoplasm. It'll start chewing on enzymes, breaking down organelles. You get the idea, right? So what do you do if you take some in? You stick it in a peroxisome. Annika, you had a question.
Okay, so like I know you mentioned lysosomes and peroxisomes. What colors are those like? Because like sometimes like on like the cell diagram, they are typically like represented by like color, like their circle. What are they typically like look like? Because like I was able to figure out the cell diagram and like it
Oh,
was
on the cell diagram,
Yes.
they're the little yellow color.
the yellow ones. Which one's that? The lysosomes, just the yellow. Okay. Are they like the fully yellow or just the yellow outline?
The little yellow, I'll show them to you when we get in there. You've already done the goal for the exercise, I can tell. You're asking very good questions. That means you've looked at it and got an idea of what this all looks like.
So, sorry.
Yes.
So like the reddish orange looking, you can call them enzymes because that's what they look like to us. Are those the peroxisomes?
Mitochondria.
Those are also mitochondria.
We'll see it when we get in there.
Okay.
Okay.
Does the mitochondria in that model look kind of like a reddish brown bean?
Yes.
Well, like,
yeah.
some of them like you could like see on the inside, and then the other ones are just like fully closed.
That's just so you can see the internal structure.
Oh, okay.
The internal structure is important because you see how it's folded? That's to increase the surface area,
Okay.
and that allows more enzymes to make more ATP. Again, this isn't a cell bio class. It is certainly not biochemistry. I am not going into the Krebs cycle,
Okay.
for which we're all grateful.
Taste the recipe.
Of course, you don't have to explanation. This is not jail.
That's high school.
I will, huh?
I said that's high school.
You are allowed to come and go. The only thing I would ask is that you try not to disturb the class. Sometimes you're going to be late. I understand. That happens, no problem. You come on in, you get set up, find out where we are, and you keep right on going. Yes, sir. Yes, ma'am.
Where is the paroxysms on the, model?
You probably won't see any on the model.
Oh, yeah, I was looking.
I'm going into a little bit more detail in the lecture than I do in the lab. One of the nice things about anatomy and physiology is the breadth of the subject. You get to talk about a lot of cool things. One of the frustrations of the subject is that you get to talk about a lot of cool things because you can't go into detail. Like telling you all about the internal structure of the mitochondria. If this were a cell biology class, you would never want to hear as much as I talk about that stuff. But this isn't cell bio. Does that help you? I just want you to know that they're two different things because you don't just have one generic stack, you stick everything in and break it down. They're specialized. Not every cell will use them. White blood cells use lysosomes all the time, right? Skin cells, probably not so much. The last one is the plasma membrane, or the cell membrane. If you call it the cell membrane on the test, I am not going to ding you points. I try to always use proper terminology and let you know why, so you can learn it. But again, we're not getting time on this, right? The function of the plasma membrane is to separate in from out. The inside from the outside. Is that important? Yeah. Can you imagine what it would be like if that wall and this wall weren't here? There's another classroom right over there, isn't there? There's a hallway that people are walking up and down. Do you think it would be a bit distracting? We only want people in here who are in this class because it helps us learn. The plasma membrane is essentially like the wall and the door. Do I control who has access to this room? I have a key, don't I? The door has a lock. I think only 2 or 3 of you would know that because only 2 or 3 of you get here early. But you've seen me unlock the door. Could I be malicious and lock Kylie out?
Of course, I could do it too.
Any one of us in here could do it, right? Because you want to control access to what goes in and out of the cell. Make sure you get that down. That's the only function that it really has, is controlling access. You want nutrients to get in. You want waste to go out. Sometimes you may have to expend energy. Everybody knows what diffusion is, right? Diffusion is simply moving from an area of high concentration to low concentration. You go from high to low. If I were to open a jar of some stinky, smelly substance and set it right here on the desk, who's going to smell it first? Probably. You'll be one of the last to smell it, won't you? Because of distance. It's high concentration up here, it's low concentration back there. Eventually, assuming we didn't have the heating system on, we didn't open the windows or doors, eventually the room would just smell bad all over, wouldn't it? Everybody see that? That's called passive transport. Passive transport. It doesn't cost the cell any energy. The stuff wants to come in. Contrast that with active transport. let's say I have more of whatever it is that I want here. Let's say sugar. I've got more sugar right here in the cell than I do outside. I don't want it diffusing out, do I? Because I want to hang on to that. I might even want to go out and grab some more and pull it in. So if I wanted more students and I creeped out there and just started pulling them in, I have to expend energy, don't I? Active transport. Does everybody see how that goes? And finally, we have another variation called semi-active transport. Semi-active transport is odd because you have a higher concentration outside that wants to get inside. Well, it seems like diffusion would be the way to go, wouldn't it? But unfortunately, whatever that substance is, it cannot just pass through the membrane. It needs some help. It might be too big and you have to use a protein gate to get it in. Imagine if that whole wall was a garage door, I could drive my little vehicle in here, couldn't I? But only if the door is up. I'd have to get one of you all to pull it up. See where I'm going? That's semi-active transport. It could also be where you have an ion that you're having to pump in. Again, it's going down the concentration gradient because it's a lower energy state, but you have to open the gate to let it in. Is everybody clear on those three? And that is the function of the plasma membrane. Section 3.2 does a really, really good job of describing the structure of the plasma membrane. I would suggest to you that you read through that, but I'm not going to question you on the exam for the detailed structure or something like that. That just kind of goes again, it's a little deeper than we have to know. But if any of this was the least bit confusing to you, the book does a good job of showing you and it's describing it. As always, if you have questions, I'm here. All right, let's see. Your book calls it facilitated diffusion. That's semi-active transport. And next up, let's talk about the cell cycle. The cell cycle. We've already touched a little bit on differentiation, which is how cells become specialized. And every single cell in your body follows a very simple 4-stage cycle. And the easiest way to envision this is basically to draw a circle. I like to kind of make it like a clock. And we start between the M and the G1. The G1 is the growth 1 or gap 1 phase. Growth 1 or gap 1. This is basically business as usual. This is where the cell does what the cell does. In a lot of cases, once the cell has become fully specialized in G1, it will never divide again. It stays functional until it dies. If that's the case, we say it is G0 to indicate that it's a fully differentiated cell that will never, ever undergo division. Everybody clear on that? That's why I drew the G0 out there. That's supposed to be a subscript 0. At a checkpoint that comes, the cell will have 2 options. One is to stay where it is, G0. The other one is to go for division, go for reproduction. So it now enters the S phase. S stands for synthesis. Synthesis of DNA. This is where DNA replication occurs. Everybody with me? Once we have gone through replication, we now enter the G2 phase, the G2. And G2 is basically growth. It gathers everything it needs to make sure that when it does divide, you have 2 fully functional cells. Do we have enough protein made? Check. Do we have 2 different sets of DNA? Check. Do we have enough organelles for each cell? Check. You see where I'm going with all this? Once it has accumulated sufficient resources, it heads into M phase. M stands for mitosis. M-I-T-O-S-I-S. Mitosis. Not your tosis, but mitosis. Haley's like, I can't believe you made a joke that way. Yeah, when I'm in pain, I do strange things. Is everybody with me? And now we're back to the start because we have how many cells? 2. And we keep going. Does everybody see that? Yes, sir.
What happens if, like, in the G2 phase, whenever it's gathering everything, if it doesn't gather everything and it tries to split?
Then eventually it will die.
Both cells?
Yeah. Well, there's only one cell.
Well, it's trying to make 2.
There's a condition here called apoptosis. A-P-O-P-T-O-S-I-S. Apoptosis. Does anybody know what that is? Cellular suicide. Yeah, I deliberately went with an inflammatory term because it helps people remember it. Programmed cell death, essentially.
Is this like the same thing as like metaphase, anaphase?
No, we're getting to that.
Oh,
Mitosis has all of those
all those
phases.
in it, okay, that's.
Apoptosis, what is the advantage of it? Why would it do that? Why would a cell willingly divide? Or not divide, but kill itself? Has anybody here ever gotten a sunburn? Got your skin peeled?
Yeah.
Reason your skin peels after a massive sunburn is because of apoptosis. The nuclei in those cells have received so much UV radiation that has contributed to so much cellular and genetic damage that rather than continue dividing and running the risk of mutations and so forth, the cells just die. There's too much damage to repair. So does everybody understand apoptosis now? Basically, whenever you get a cell that is no longer functioning, kind of like what you were asking, it enters what we call senescence and then apoptosis. Senescence is basically just a fancy word for cellular aging.
So whenever you said when it gets to N, it like goes back and cycles again. So once it's already divided, will it do that again? So does it just keep on?
Until we hit the Hayflick limit.
Or it's a cancer.
Or if it's cancer, or if it's an embryo. I did talk about the Hayflick limit, didn't I?
Yeah. The max amount of times the cell can.
I didn't go into telomeres or telomerase.
Yeah, you did.
Okay. I just have to make sure because I have
That's
so
when it,
much
like, cuts off
overlap,
a
I don't
little
want.
bit of the DNA every time, right?
Yeah. Just chops off a little bit of the DNA, and each time it gets shorter and shorter. Eventually you hit something that's important and you're just dead. But that is just a fact of life. I mean, none of us could exist without oxygen, could we? Yet, believe it or not, the very fact that we use such a reactive chemical as oxygen in our bodies is probably what ultimately causes so much cell damage that we end up dying. But it's the only way to get all of that. Okay? Life is pretty amazing. You'll find living things everywhere on this mud ball that we call home. We are infested with life. That's a good thing. It's one of the reasons I like this. But eventually, pretty much everything will die. Okay. Now to Kylie's question. She says, is that the same thing as mitosis? Yes, it is. A lot of it depends on whether you're a cell biologist who like to look at things like this, or you're a geneticist who likes to focus on M. If we were doing this, to break it down, everything between these two phases would be called interphase. Geneticists don't care about interphase. That's where the cell is doing its normal thing and growing, right? But we're not interested in the cell doing its normal thing. As geneticists, we're only interested in the DNA. So, mitosis is reproduction of the nucleus. Reproduction of the nucleus. What goes on in mitosis? Well, you've got 4 steps involved. At the very beginning of it, you have 1 nucleus. At the very end of it, you have 2. Is everybody clear on that? First step is called metaphase. Or excuse me, prophase. Prophase. Tell I'm a geneticist, I get right to the middle of it. When you look at a chromosome, or when you look at a cell, you normally don't see the chromosomes. They are normally in a relaxed state. The DNA is open, it's transcriptionally active, meaning enzymes can access it, it can make messenger RNA copies. That's a good thing. But when prophase starts, you're going to pack the DNA or condense it. This is where and when the chromosomes become visible.
And this is all in mitosis, right? This is all part of mitosis.
Yes. Prophase, they pack up, they condense, and now you can see them. How efficient is the packing? Well, here is a little tidbit that might blow your mind. If you took all of the DNA in an average human cell, and you put it together in one long chain, and you unwound it and unpacked it, it would be 6 feet in length. I'm a little over 6 feet tall, so imagine a strand of DNA my size packed into every single one of your 50 trillion cells. People ask me why I like genetics. DNA is cool. So it packs it down, right? Then in metaphase, all of the chromosomes line up in the middle. They line up in the middle.
What phase was that?
Metaphase. PMAT is how I remember it. Prophase, metaphase, anaphase, telophase.
We learned it a little bit in AP biology.
In biology, I go into it a lot more deeper than that. Also, during prophase, the nuclear membrane is beginning to break down, and you're building a structure called a spindle. You can just imagine kind of a basket. And all of the chromosomes are lining up right where my fingertips meet, right there in the middle. Everybody see that? Metaphase is by far the easiest to see under the microscope. You look for that conga line lined up right in the middle, those deep, dark chromosomes. Now, the sister chromatids begin to pull apart and again to pull towards the end. Does everybody see that? We call this anaphase. Anaphase. A-N-A-P-H-A-S-E. Anaphase is just the movement of the chromosomes to each end. It is followed by telophase or telophase, T-E-L-O-P-H-A-S-E. Telophase is basically the opposite of prophase. Instead of the nuclear membrane breaking down, it starts to reform. The spindle, instead of being built, is being broken down. The chromosomes begin to decondense and go back into their relaxed states. And when it is done, you have two nuclei. Now notice I did not say that the cell divides, did I? That is because in some cases, the cells in your body contain more than one nucleus. Muscle cells. Didn't know that, did you? Why would you want more than one nucleus? Well, imagine if I gave you all a sheet of paper that you had to read between now and next Tuesday. And the only copy was kept over in my office. Be a bit of a problem, wouldn't it? Because there's about 20 of you and only one sheet of paper. If I gave it to every professor on the second floor of Stoner Hall and every one of them had their copy and it didn't matter where you went, it'd be a lot faster, wouldn't it? So it's efficiency. So generally, along with mitosis comes cytokinesis. Cytokinesis. Cytokinesis is the division of the cytoplasm. But I want to emphasize that you can have mitosis without cytokinesis on occasion.
You can.
You can. That's how you get multinucleate cells. Yes, sir.
What's cytokinesis again?
You can have cytokinesis with mitosis, but not without. You can have mitosis with or without cytokinesis. Questions?
So with going back to like mitosis, I'm looking at a diagram of like what you're saying, and like it points out the centrosomes. How and it shows that there's two sets of centrosomes and that they're splitting. But how whenever they do split, each gets one set.
Yeah.
How many say those two daughter cells, can those after it starts with the one and it has the two daughter cells, can those two daughter cells then technically split?
Because of S phase.
Okay.
You have in your body two sets of DNA.
Mm-hmm.
You got one full set of chromosomes from your mother, 23. Everybody agree? You also got a full set of 23 from your father. So everybody looking and seeing them on each two sets, right? During S phase, the DNA replicates. That means it copies itself, right? How many sets do I have now? 4. Everybody see that? These two are identical, aren't they? These two are identical, aren't they? We separate, we have how many cells? 2. Each has how many? 2. If they go through mitosis, they'll double and divide.
Okay. So there's not a set number of centrosomes you can have, they just keep those as well, keep dividing?
Until you hit the Hayflick limit.
Okay.
You might be thinking of meiosis, which is how you go from 2 sets down to 1. Why would you only want 1 set? Reproduction, right? So what happens when gametes are made? They go through a variation of this called meiosis. The rule for mitosis is to double the DNA, then divide. Double, then divide. The rule for meiosis, if you're wanting a single cell with 1 set, it just seems like it would be easiest just to divide, right? But unfortunately, evolution doesn't work that way. So it's double, divide, then divide again. It's actually 2 stages. And based on what you're asking, I think that's maybe what you're thinking about.
I'm just looking at a diagram and it's pointing out the centrosomes and it's.
The centrosomes are just the organization point for the spindle.
Yeah, that's okay.
And I apologize if it seemed like I'm not going deep enough. The real challenge is to make sure I don't dive too deep and lose all of you, because we don't really have to go to that level. I mean, do I want you to know the cell cycle? Yes, that's almost a guaranteed test question, isn't it? Draw a diagram, draw and label the steps of the cell cycle, or list and describe the steps of the cell cycle. Do you need to know mitosis? What do you think? Yeah. Do you need to know the organelles that we talked about? That help? And it looks like we are out of time, so I will see you all in the lab in 15 minutes.